Thank you, and thank you to the organizers for inviting me to join you today. It's really a pleasure to be here. I didn't know we were going to have a little North Carolina contingency. So today I wanted to talk about our project and looking at the use of whole exome sequencing in newborn screening. But in order to do that, I really wanted to give you some basics. I know some of you are probably genetics experts in this room, but others may have very little knowledge of basic genetics. And I think when we talk about some of the new tools that we're able to use in genetics and genomics, it may be helpful to go over some of those things. These are some of the objectives, hopefully, that things that you'll have a better understanding of at the end of this talk. And some of the definitions, genetics just means determined by genes, whereas the genome is really the complete DNA sequence of an individual that contains all of their genetic information. And genomics is the field of genetics that's concerned with structural and functional studies of the genome. So just as a bit of a refresher, if we talk about a cell in the body, any cell except the red blood cells, so any other cells that contain a nucleus contain DNA or genetic information. That DNA is arranged in structures that are called chromosomes. These are tightly wound pieces of DNA in proteins. And then if we were to stretch it out, you'd see the typical double helix making up the DNA. So chromosomes, if you're familiar with cytogenetics, is what this would look like, would represent the chromosomes from one cell in the body looked at under a microscope. And what we do is arrange the chromosomes in order of their size and also based on what's called staining or banding, that is produced when we put a special type of stain on the chromosomes. So they're arranged in order from the largest down to the smallest, and they're in pairs. Chromosomes 1 through 22 are called the autosomes, and then we have the sex chromosomes, the X and the Y chromosome. The chromosomes you can think of being made up of genes on a long string, and each chromosome contains 1,000 or more genes. Now, DNA itself is made up of molecules that are termed nucleotides, and these are the four that you see here. You can just remember A, T, G, and C. And each nucleotide has a corresponding partner that's called its base pair. Now, a gene is made up of thousands of these nucleotides or base pairs. And genes range in size from small ones that are only about 250 base pairs to very, very large ones, like the gene for muscular dystrophy, Duchenne muscular dystrophy on the X chromosome, which is 2.5 million base pairs in size. So if we look at all the letters making up that gene, there can sometimes be differences or changes in one of those nucleotides. So for example, if we look at the C, and if that is changed to a T, that can be called a gene variant, okay? Now, gene variants are very, very common. We all have about 3.8 million different variants from one individual to the other. And most don't cause any problems at all. They're just common variations. And they're also known as polymorphisms, which typically we mean by that is that they're present in 1% or more of the general population. Most are benign, don't cause any disorders at all. However, some are not benign and some can cause problems, either with the protein product that gene produces or with some regulatory or control over the gene. So really one of the trickiest things in genetics now is figuring out what's just a benign variant and what is a pathogenic mutation. Sometimes you'll see the term mutation meaning a deleterious variation. Other times, a mutation, the term may be used as also a benign type of change, and they'll say like a deleterious mutation. So you have to be a little bit careful about the use of some of these terms. When we look at changes in a person's DNA, one of the main things that we want to try to figure out is if that change is deleterious or is it just a benign polymorphism. One of the resources that we have to use for that purpose are different databases. And as I'll talk about later, oftentimes these really aren't as adequate as we would like. It's certainly an area where we're trying to gather more information all the time, and in time we'll be at a stage where it'll be a lot easier to figure out what's benign and what's deleterious, but right now that's certainly, one of the big challenges that we have. So looking at this in a way that I think makes it a little bit easier to understand, about these types of mutations, if we think about a sentence that normally should read, "The cat saw the dog," there are certain changes that are at the base pair level. So you're substituting one of those letters, right? An A for a C, for example. And in this case, if we're substituting a B for a C, it changes that meaning of the sentence. The bat saw the dog, and that would be equivalent to a point mutation. If we took out a word and took out the saw, the cat, the dog, is similar to what you'd see in a deletion. An insertion type of mutation, the cart saw the dog, we put in an extra letter. And then there are certain types of genetic variation that are triplet expansions or various types of expansions in genes. And this would lead to the cat saw the dog. Similarly, if we look at this at the nucleotide level, you see the same things here: a point mutation, a deletion in a few of the nucleotides, or an insertion, some extra nucleotides. When we think about different types of genetic testing, these range from diagnostic testing, which is a clinical geneticist I'm most involved with. We see patients with multiple congenital anomalies, for example, or intellectual disability, autism, and we run different diagnostic genetic tests to try to figure out what the underlying problem is. Also, you've heard about diagnostic testing, genetic testing for cystic fibrosis. We can also use diagnostic genetic testing for breast cancer or colon cancer. Genetic testing is also used some in screening. In newborn screening, you've heard about its use in cystic fibrosis as a second or third tier of testing. Also for example, prenatally, when individuals go in for preconception counseling or prenatal counseling, they're typically offered carrier screening for things like sickle cell and cystic fibrosis, and depending on their ethnic background may be offered additional testing such as the Jewish genetic disorder panel that includes Tay-Sachs disease. And then we also do predictive testing or pre-symptomatic testing for individuals who may have a family history of Huntington disease and want to know if they have inherited that gene from an affected parent. Also, breast cancer. If a woman has a family history of breast cancer or she herself has breast cancer, is often offered genetic testing for the breast cancer genes. And then another area where genetic testing is being used more and more is in pharmacogenetics and individualized health care to try and determine what medications may work better in some individuals than others, also helping to know the correct dose of something like Coumadin used for anticoagulation to find out the appropriate dose based on the genetic results from a patient. And genetic technology is also used in what are called GWAS studies, genome-wide association studies, where they're really using these polymorphisms in different parts of the genome to try to figure out genes for things like diabetes and other, common disorders, hypertension, things like that, are based on GWAS studies and linkage analysis. And then you often read about genes for different traits. And that still certainly has a long way to go, but things like musical ability, artistic ability, someone's found a gene that may be associated with things like that. But there are many many ethical issues that arise in genetic testing, and especially in pediatrics, where we think about that a lot in our research studies. Genetic testing is different in a number of ways. It has implications not only for the patient that you're testing, but also for other individuals in the family often. And in thinking about genetic testing of minors, what are the unique issues that arise when we're doing genetic tests or offering genetic tests in children? Thinking about privacy and discrimination issues. In 2009 or 2008, the law called GINA, the Genetic Information Non-Discrimination Act, was passed. And this is to prevent discrimination in health insurance and employment based on genetic information. But GINA does not include members of the military who can suffer discrimination based on their genetic testing results. And also it does not prevent other types of insurance, such as life insurance, disability, and long-term care insurance from being denied because of genetic test results or genetic information from an individual. So despite all that we've learned about, genomes in humans, there's still a lot of issues that I think we need to address. I've already talked about, chromosome disorders as one way of really looking at a person's entire genome. We couldn't look too closely, though, because I said every chromosome is made up of thousands of genes. And I like to use the analogy, and Angela Shirley I don't think is here, but I took this from a presentation that I heard her give a few years ago because I thought it was a helpful analogy to make. But you can sort of think of being able to look at chromosomes like a Google view of Earth. And we're able to look down at a neighborhood and see maybe, how many houses there are, but you really can't see too much more than that. So there's a lot of information there that we're still not able to see. So through the years, we came up with a better way to look at a little bit closer view of the chromosomes, and that's called microarray or cytogenomic array technology. And this detects copy number changes, so it can tell you if there are a number of those base pairs or nucleotides that are missing or extra, which is certainly helpful. And so because of that, we're able to diagnose conditions in individuals with, let's say, intellectual disability, find out the causes of that we never could see on a routine karyotype. So it's really enabled us to look at the entire genome, but with a much higher degree of resolution. But it can't detect very small changes at the level of nucleotides themselves. Most of the commercial microarrays that are used have probes that are spaced about 1,000 base pairs apart. So anything smaller than that, you're not going to be able to pick up with the microarray. Coverage does vary. Some parts of the genome, there may be much closer together probes, and others are much farther apart. It's very difficult to see any deletions within genes themselves, although there are some specialized microarrays that are enabling people to do that. So anyway, but the important thing is deletions or duplications or those base pair changes, like those single letter changes in the genes that we talked about, we wouldn't be able to pick up with the microarray. So again, using the analogy of looking at a neighborhood, well, we can see a mailbox and see how many pieces of mail are in that mailbox, but we really can't get down to whether there is, a misspelling or, what's printed on one of those, a letter or a bill in a mailbox. So in order to do that, we really have to be able to read the letters of the genes. And we do that through a methodology that's called sequencing. And sequencing is just really reading those individual letters one by one. We use different preparations in order to do that. So we have a reaction mixture that includes a way to label each one of those nucleotides. So again, that CGAT is included there. And one of the original types of sequencing was invented by Dr. Sanger, who passed away, I think it was last year. But anyway, it's commonly known as Sanger sequencing. And the readout is, as you see here, so that you can see if there is a change or mutation in a gene. This also has been termed first-generation sequencing. So now we've skipped ahead to what's called next-generation sequencing. And I like to use the analogy of basically being able to read the whole encyclopedia, having the whole encyclopedia in your mailbox. This has also been called massively parallel sequencing. And it's not that different from Sanger sequencing. It's just that it has to go through a lot more different reactions and technically is more difficult. But basically, you're reading those letters, the A, T, G, and C again, and utilizing a reference sequence that's compared to. And depending on the depth of coverage that you have, because you're not going to really get an accurate reading with just one pass, you're going to have to look over and over again to see if a change that you're picking up repeats itself, indicating that there truly is a variation or possible mutation at a specific point in that gene. But it can theoretically search for mutations in all of the human genome, so about 20,000 genes. There are a couple of different methods used. The most commonly used one, especially being used now on a commercial basis, is called whole exome sequencing. And the exome is made up of our exons. And the exons are really the working parts of genes. They're the parts of genes that code for proteins. And whereas the whole genome is everything, all the exons plus all the stuff in between all those exons or the introns. But the analysis of this is still extremely complex. And again, our understanding of what's a significant mutation and what is a polymorphism has a long way to go. And this is just from a recent article that was published in the New England Journal of Medicine by Les Biesecker and Robert Green from the NIH, and looked at some of the basics of utilization of next-generation sequencing and had a nice comparison of exome sequencing versus genome sequencing. The raw data really looks like this. These are called band files that come out as a result of whole exome or genome sequencing. And again, some of these may have some changes, but they're not seen in all of the reads. And the depth of coverage, which can range a lot depending on the type of sequencing method that you use, varies quite a bit. If we look back to contrasting Sanger sequencing, our traditional sequencing, with whole exome sequencing, Sanger can generally, running a Sanger sequence can give you about 100 to 800 base pairs. It's very good for targeted mutation analysis if you know exactly where to look at and which gene to look at. And it gives you very good coverage of that entire gene, and it still remains the gold standard for sequencing of a gene. Whereas whole exome sequencing, as I said, can be 30 megabases of genetic information that you're looking at, or if you do whole genome, that's 3 billion base pairs. It carries with it increasing problems with interpretation, and it's still not considered reliable enough to be a standalone test. So if you're really going to use this for diagnostic purposes, you always want to confirm it with Sanger sequencing to confirm that the information is accurate and correct. The use of whole exome sequencing on a clinical basis has been shown to be very effective. This was a paper published last year by the group from Baylor about their experience with doing clinical whole exome sequencing for the diagnosis of Mendelian disorders. And they found that in patients who had had other routine types of testing, such as karyotyping, chromosome or cytogenomic microarray testing that was all negative, and when they did whole exome, they found that in about 25% of cases, they were able to come up with a specific diagnostic answer for those patients. One thing that also comes up, because we're looking at so many different genes, is the concept of what are called incidental findings. So things that you really didn't set out to look for, but which, as a result of looking at all the genes, you do find out about. And with next generation sequencing, these can include genes, for example, for breast cancer. So maybe you're doing a whole exome in a child with developmental disability, but you find out that, oh, by the way, that child also carries the gene for breast cancer and may have a 80% or higher chance of ultimately developing breast cancer when they're older. Or things like cardiac arrhythmias, for example. So what information should be reported back to patients, and in the case of children, to their parents, when we're doing tests like that? And so that's taken up a lot of interest and time over the last several years as this technology has become more common. So now I'm going to switch over and talk about how use of whole exome in newborn screening is something that we're starting to look at now. So as I'm sure most of you know, newborn screening is a public health program aimed at the early identification of conditions for which early and timely intervention can prevent or reduce associated mortality and morbidity. You heard this morning about the recommended uniform screening panel based on recommendations from a 2005 task force that was convened. And you've heard about how new conditions can be nominated. There are now 31 core disorders. And if we look at some of those conditions that the panel or the experts initially looked at, there were quite a few that didn't make the cut because there were no tests available for them. So conditions that people felt, were probably, somewhat reasonable to look at, but well, we can't pick them up with tandem mass spec, for example, or any other, known laboratory test at that time. And so those that were excluded, about a third of all the ones that they considered were excluded because there was no test available. So now with the use of sequencing and next generation sequencing, theoretically barriers to adding any disorder to a newborn screening panel can be overcome if there's a known genetic etiology for that condition. However, as recognized early by Wilson and Youngner in thinking about screening, the central idea of early disease detection and treatment is simple. However, the path to its successful achievement, bringing to treatment those with previously undetected disease, and on the other hand, avoiding harm to those persons not in need of treatment, is far from simple, though sometimes it might appear deceptively easy. And I think with all these things in mind, the NIH in 2012 invited applications to explore the implications, challenges, and opportunities associated with the possible use of genomic sequence information in the newborn period. Because we had a background in looking at one specific condition, Fragile X syndrome, in newborn screening, we thought that we were in a fairly good position to apply for the grant. And fortunately, we were one of the 4 centers in the country that did receive funding, along with Brigham and Women's Hospital and the Harvard Group in Boston, Children's Mercy Hospital in Kansas City, and the University of California at San Francisco. We're all kind of looking at different areas of this. And in thinking about next generation newborn screening and some of the things that we're working on is, can we broaden the definition of what's treatable? Maybe things that could be beneficial to detect early might not really be treatable in the traditional sense. What about extending the concept of benefit of screening to not only the child but to the family? But how do we do this while still protecting the autonomy of the child and balancing the rights of the parents to have information? And what about pre-symptomatic genetic testing in children? And then these incidental findings that we're going to come up with if we do next generation sequencing. In our Fragile X newborn screening project, we found that when parents were given the option to participate in our study, that about 60% or so did accept it and wanted to have their children screened. Again, totally on a voluntary basis and with informed consent and brochures that gave them all the pluses and minuses of having testing. More recently, there was an Australian study that looked at newborn screening for Fragile X, and they had a much higher rate of acceptance. 94% of parents accepted that, so that this suggests that culture and context play a role. Our whole exome sequencing for newborn screening project is called NC Nexus and is funded by NHGRI and NICHD. And our overarching aim is to evaluate whether next generation sequencing can extend the utility of newborn screening, to devise a clinically oriented framework for analysis of next gen sequencing newborn screening, and to develop best practices for trying to incorporate it into clinical care. Some of the specific aims include what we call our binning committee that looks at an analytical strategy for determining how and whether incidental findings should be reported back to families. This is an overview of what's called the pipeline for analysis. And the main thing I wanted to point out is that the actual sequencing is a relatively small part of the whole thing. The variant calling, using databases to help determine what's a pathogenic or non-pathogenic mutation, the annotation, when the machine or the pipeline gives us information, then humans have to sort through all of that information, go to the literature, look at what's known about those mutations. Are they true mutations or not? So whereas the sequencing can be done in a day or two, it's all of the analysis part that really takes so much time, that can take 6 or more hours per patient. And then meeting with the families, things like that, which needs to be done very carefully and with a lot of pre-test and post-test counseling. I think I've already covered most of this in terms of what are different, how many different variants an individual would have. And so basically this pipeline slowly narrows down all these different possibilities to try to give us what would be a deleterious mutation or not, because certainly we don't want to give inaccurate information back that oh, your child has a genetic mutation when it's just one of these normal polymorphisms. And there's really no gold standard method for doing this, again, hopefully with time, and there are a lot of projects going on at the national and international levels, but hopefully it'll get better in the future. So when we think about what we would include in a next-gen newborn screening test, these would include conditions that are currently on the recommended uniform screening panel, but also conditions that would fit in but maybe weren't part of the RUSP because they didn't have a test available. And so along with my co-PI, Jonathan Berg, and previous work that he'd done with Jim Evans at UNC, they developed an algorithm to think about each gene individually and to do scoring based on the severity of the condition that gene is related to. So does it cause sudden death, serious morbidity? What's the likelihood of people, if a person would have a mutation in that gene, what's the likelihood that they would develop that problem? Is it very high, greater than 50%? Or for the most part, are people unaffected or less, very unlikely to be affected with that condition? What's the efficacy of knowing about this? Is there screening available? Is there treatment available? Is it highly efficacious to know about this early on in a person's life? Or is it pretty ineffective? And then how acceptable would it be if we're talking about, let's say, breast cancer gene for mammography that would seem to be minimally, moderately to highly acceptable for most individuals. But there are other things, for example, familial risk of stomach cancer, for which the only real known treatment is removing the stomach. And that certainly would be very unacceptable to most individuals. And then what's the knowledge base? There's some conditions for which there might be one published paper out there. There are other conditions that are much more common, and there have been many papers published. So the scores come out to anywhere from 0 to 15, and it's really that score that we are hypothesizing determines the medical actionability of that gene. And to give you an example of something that is part of newborn screening, if we look at the gene for phenylalanine hydroxylase that in homozygous form or compound heterozygous form, two mutations would cause PKU. If we, based on the scoring method, the severity of the disease was given a 1, which I think some people would argue it's not life-threatening, but certainly causes serious morbidity. But anyway, it came out with a 1. The likelihood of that happening in an untreated individual, very likely, greater than 50%, got a 3. We know that there are very effective interventions, so it got a 3. The diet, the special diet, not the easiest thing for a patient to have to do, but not the worst either, so it got a 2. And then certainly we have a lot of information out there about the effectiveness of treatment, and so on. And so the knowledge base got a 3. So that ended up with a total score of 12. Then if we look at the gene for a condition that can cause colon cancer, familial adenomatous polyposis, the severity score was a 2, possible death due to cancer, highly penetrant, likelihood of developing multiple polyps that would become malignant was a 3. Colonoscopy is a very good screening tool, so effectiveness of intervention was a 3. And the acceptability of intervention, at least yearly colonoscopies, got a 2, and the knowledge base was felt to be high, and so got a 3, and that actually got a bit higher score, a total score of 13. So looking at this in terms of the actionability, degree of actionability, and then the onset, we come up with this age-based modified metric system that we're using. And if you look at conditions of early onset, so childhood or infancy onset conditions, things like MCAD and PKU, for example, that would be in the upper left quadrant, and that would really make up what we are terming the next generation newborn screening. Then there are adult onset medically actionable conditions, things such as Lynch syndrome, another type of familial colon cancer, and breast cancer genes. So not something that we'd be concerned about in children, but that would, be important to know about in adulthood. So these would be of adult onset medically actionable conditions. And then there are those that would have early onset in childhood, but for which there's no known effective treatments right now. And so these would be childhood onset non-medically actionable. And then the conditions of adulthood, such as Alzheimer's, for which there's no known treatment at this point, and these would be the adult onset non-medically actionable. So as hopefully in the future treatments get better for some of these conditions at an earlier age and are medically actionable, they'll sort of shift to different quadrants. Likewise, if an effective treatment for Alzheimer's is developed that, maybe starting at a very early age, that would also shift the quadrant that would be in. So just a way to sort of look at that. So that's a big part of our study is, going through the different genes, and we have a large committee made up of our genetic counselors and clinical geneticists, metabolic dietitians, ethicists, anthropologists, and so on that are part of that committee and all weigh in as we look at these different genes. In the clinical aspect of our study, we are looking at the sensitivity and specificity of whole exome sequencing. And so we're going to be using patients with known conditions and also those with conditions that are currently screened for in standard newborn screening, and also conditions that are not currently screened for, but which we think, might be helpful to know about at an early stage. So we have a cohort of children with PKU and MCAD, cystic fibrosis, and hearing loss, and then another with some of the other conditions that have been recommended for newborn screening, including the mucopolysaccharidoses and adrenoleukodystrophy. And then we also are studying a group of well children whose moms will be recruited during their pregnancies, and we're interested in looking at, the uptake of wanting to have this testing done on children who are otherwise hopefully a healthy group. We're hoping to be able to do the analysis from saliva samples from infants instead of having to get blood samples from them. And we'll invite families to participate. We'll have 200 in the diagnose group and 200 in the total, in the well-child group. And then we'll randomize the subjects in the study so that everyone will get the next-gen sequencing newborn screening results. And then for that additional information, so for example, those conditions of either early childhood onset but that aren't treatable, or adult onset that are treatable, they'll have options of getting that information or not. So some will be randomized into what's called the choice group, because we're interested to see how parents deal with choices like that, whether they want the information or not. And hopefully we'll also be able to report back carrier status for recessive conditions, or give that as an option to parents who are in the choice group. And then we won't be reporting back any information on adult onset non-medically actionable, such as Alzheimer's. The third part of our project is examining the ethical, legal, and social issues of doing next-generation sequencing in children and newborns. I've already talked to you about the binning, or determining categories of genes, but we'll be getting input from parent groups as to how they think about different genes and what information they want to know. We're developing an online decision aid tool that hopefully can be utilized by others in the future to help parents think about whole exome sequencing and return of results. We've had an external advisory committee made up of individuals with expertise in newborn screening and ethical issues regarding testing in children. We are surveying different stakeholder groups, having focus groups, and then, as I said, are developing this decision aid tool to help in the return of results part of the information that we'll get. For the affected cohort, we'll report back both pathogenic variants and variants of unknown significance in genes related to their underlying condition. So we expect a high rate of positive findings in that group because they have, known conditions. We also will report back other information regarding these other conditions that we've put into that upper left quadrant category, but those will only be definitely known pathogenic results. And then the same will go for the healthy newborn cohort. They'll get the known pathogenic results. And then the randomized group that can decide if they're randomized into the decision part can decide which additional information on which genes they want to know about, if any. There's certainly an option that they won't get any of those additional findings back. Certainly, follow-up in newborn screening is something that my group is very involved with at UNC, and we know that the public health benefits of testing and improved outcomes through newborn screening programs will only be realized if there are adequate resources for long-term follow-up care and management. And as Susan Berry has said in the past, newborn screening is only as effective as the care it prompts. And as we think about next generation newborn screening, it's really going to be a real challenge to make it beneficial for patients. So when we look at some of these different questions, can next generation newborn screening expand the utility of newborn screening? If we can test for some additional conditions, such as I mentioned, ones like early colon cancer in children, FAP gene, multiple endocrine neoplasia, type 2B, that can lead to very early thyroid cancer. We've had a number of patients over the years at UNC, young children with metastatic thyroid cancer. If we could find out those children who are at risk for that ahead of time. Some of the primary ciliary dyskinesias that can lead to multiple respiratory infections and chronic illness in patients. And also, can we improve the specificity and sensitivity of the screening that we already do, that we know is very accurate, but for some of the screening that we do in newborns, such as, I became interested in this really because of an interest in hearing loss and the etiology of hearing loss. And almost every set of parents who I meet with whose child has been diagnosed with congenital hearing loss are worried that their child has Usher syndrome, which is very, very rare, but a condition that not only is a genetic condition that causes hearing loss, but also will lead to vision loss, severe vision loss, and blindness in childhood. And so, whereas it's very helpful to know about the hearing loss in the child, we're raising the anxiety level for their families because they think, unless we can identify what the cause is of the hearing loss, they imagine, the worst case scenarios. And also for some of the other conditions that we deal with, like PKU, would there be a way that knowing the specific mutations in the gene and whether or not they might correlate with things like use of Kuvan, some of the other interventions that are being used for patients, for the severe combined immunodeficiency conditions. The ones that are associated with T-cell deficiency can, at least we think, be picked up through newborn screening, but those that don't affect the T cells can't be detected. Also, the newborn screening just detects that there, could be this SCIDs, but it doesn't tell you, exactly what the etiology is. And there are some of these immunodeficiency conditions where really early bone marrow or stem cell transplants are critical to do, whereas others are milder and might respond more to like immunoglobulin therapy, for example. So hopefully we'll, be able to improve the specificity and sensitivity of screening. So my opinion about all this and next generation newborn screening, it's certainly not ready to be a standalone test, nor do I really ever see it being such. I think that, as time goes by and we get more information about what genetic changes mean, that it will, be more informative for everybody. But I think it will always be utilized along with the traditional newborn screening that we're doing now, biochemical-based testing and so on. I do think that there's probably a benefit from some of these panels where you would just have specific genes that you'd want to look at. And that's what a lot of the diagnostic whole exome sequencing tests are now, that are being offered. There's just specific panels that they'll look at rather than doing a whole exome on it. And other things to look at, if we're doing genetic sequencing and we don't return those results to families, who's the keeper of that information? Is it the the newborn screening people, the researchers, the pediatricians, whose responsibility is it? And I don't have answers to that. Do parents have a right to their child's DNA sequence? What if some of these conditions that we wouldn't report out now then become treatable in the future, so we'd want to give that information out? How do we recontact patients? We've seen how hard it is to recontact patients even, a few weeks after their child undergoes newborn screening, standard newborn screening. So what about if it's, months to years down the road? Who's responsible for figuring out when new discoveries come out about genes and variations? And then what about commercial testing? And already companies, Pair-based Genomics is offering next generation screening of newborns and not as a public health test right now, but looking at like sick newborns. And actually one of the other centers funded through the U19 from NIH is looking at rapid turnaround time for whole genome sequencing in children or infants in the neonatal intensive care units, and that's the group at Mercy Hospital, the study that Stephen Kingsmore is doing. And I think definitely in a population like that, there's definite benefits of doing the whole exome sequencing for a rapid diagnostic purpose. But it is a whole different thing if we're talking about public health and testing otherwise normal babies. And should all of this be mandatory or voluntary? Some have said this could be like a secondary type of newborn screening that's offered to families. But then what about the health disparities? Is that something that then only those of means would be able to afford? And looking at it as the Secretary's Advisory Committee is, looking at the demands on public health and healthcare systems will be extremely important. And then despite the Affordable Care Act and GINA, what about genetic discrimination in terms of employment and for insurance? And I just leave you with this. This was something that came out on the internet a few weeks ago about a man who's a graduate student at University of California at Davis and decided to sequence his son's genome before he was born. He was able to get a sample from the CVS that his wife had and got the company or the commercial laboratory that did that testing to send him back some of the cells and DNA. And he worked in a lab where they were doing whole exome sequencing, and he went ahead and did it on his son. And I think he had some regret afterwards and saying that our attitude is that you make a lot of decisions for your kids, including ones that may seem sketchy in hindsight. So this is definitely something that's out there. Using it prenatally, postnatally is something that really needs a lot more study and a lot of input from many stakeholders. And I'd like to thank my co-PIs and the other investigators in our group, Laura Milko, who's our project coordinator, and the folks who make up our binning committee, the group that is thinking about all the different genes. And I'm happy to take any questions. Thanks. So the question is, if I wanted to do a study on a genetic study on a newborn, would I go to microarray first? You mean versus like a routine karyotype? Yeah, certainly, the American College of Medical Genetics, some of the other advisory groups have recommended microarray first because you will pick up more than you would in a routine karyotype. I have to tell you, at my own institution, we're still doing both. We're doing the standard karyotype. For one reason, we can get the information out much quicker on the routine karyotype. We have like a 48-hour turnaround time versus the microarray usually takes about 2 weeks to come back. But cost-wise, it probably, microarrays are more expensive, but you get more bang for your buck with the microarray. So yes, there is some reason to do that.
The other question is, if you have a patient that you're thinking about to be Fragile X syndrome, but the karyotype is normal, what will be the next?
So Fragile X is really important to keep in mind that a karyotype, a microarray, and whole exome sequencing are not going to tell you about Fragile X, okay? You've got to order the specific DNA Fragile X testing because none of the other things will pick it up. It's one of those triplet repeat expansions, and so right now, microarrays are not sensitive enough to pick it up. Karyotype, you won't see it on the karyotype in the old days, we used to do, like, we'd grow the cells up in a folate deficient cell medium, and you'd actually try to look for the little fragile site on the X chromosome, which is where it got its name from. But that's not a good way to pick it up because you miss a lot that way. So you need to order it. And I've actually seen a couple of patients recently where they've gone through this huge gamut of testing. They've had like a $10,000 whole exome, commercial whole exome done, and it's all been negative, and they never tested the child for Fragile X. And anyway, it's an important question and good to think about what you can't detect with next gen and
Thank you.
microarrays. Mm-hmm.
Thank you for a very brilliant talk. That was wonderful. My question was, do you think, I know right now it might be an expensive test, but doing this whole exome sequencing on cell-free fetal DNA would be the next thing to do?
Yeah, actually, Deborah and I were just talking about that yesterday, that I think there was at least one recent report of a group that did do that. So, taking the fetal, cell-free fetal DNA from a woman's blood, a pregnant woman's blood, to do analyses of the fetus. And it's now being used on a clinical basis to screen for trisomies, like trisomy 13, trisomy 21. But once you have that DNA, you can basically test for anything. And yes, so I'm sure that's the next thing coming down the pike would be whole exome prenatally. I mean, they're trying to do it now, I think, using microarrays and analyzing for copy number variants on free fetal DNA.
I think they're doing that multiple parallel sequencing, actually, I think. I don't know if I'm
Next
right
gen,
about
yeah.
But that would, shift the onus and the cost to before even the baby was born, so you wouldn't be like having trouble contacting people and having this whole battery of setup of people that needed to coordinate care,
Right.
because you could do it.
But then what to do with these variants of unknown significance? It's really going to make it even trickier.
Maybe tell them to deliver at a tertiary care center where there's more treatment available or
Yeah.
screening. And thank you very much.
Somebody in the back there. Oh.
Great talk. I'm just curious what the time frame is from when you collect the sample from the fetus or the baby in this hand to returning the results to the parents. Because if we're trying to intervene, in day of life 2 or 3, is that possible for some of these conditions as we're running this?
Yeah. For our research study, it's probably going to be months, based on our other whole exome study that we're doing, where it's months and months for return of results. But if you're some groups are really doing this rapid turnaround time where they're saying, within a few days they'll have the results of the whole exome. So it's going to be shortened in the future.
Great, thanks.
Yeah. Man in the blue. Oh.
What about cost?
The cost for whole exome? Yeah, so we budgeted $1,000 per whole exome in our study, but again, that's just really the equipment part of it and getting the materials and everything. It doesn't pay for all of the interpretation part of it. We do have a core facility that helps with that, and then we have a lot of people on the research study grant who are getting paid to help with that. But
So commercially, it would be much
commercially,
higher?
I think it's like 8 to 10. 6 to 10. Yeah, I mean, our hospital always adds a lot on top if they're sending it out. I'm not sure exactly what Baylor's charging right now, but I think it's like 6,000 to 8,000 or so, and coming down. There's a man in the back who's had his hand up since the very beginning. If you could please either shout out or yell.
Hi, my question, actually, a two-part question, but it's related to the last one on costs. Who's going to pay for all this going forward, and in the world of trying to manage healthcare costs, obviously outside your research and in the commercial world, and also have you personally been involved in determining what is going to be covered by managed care?
I have not been involved in that part of it. I can say, though, that, my experience seeing these kids who have gone through many, many different diagnostic workups, that have, not shown anything, that have probably cost like $20,000, and then they have a whole exome that gives them an answer, and the whole idea that you're avoiding this whole diagnostic odyssey of, trying to come up with, a specific diagnosis. So I think it can in some ways be cost effective. But yeah, I mean, I totally agree. Is it the best, use of money? But it in many cases gives you more information. You're more likely to find something than from, let's say, an MRI. And, some kids have had several brain MRIs over the years to figure out why they have intellectual disabilities.
Right, we just need someone like yourself to determine who actually needs to get that at the front end versus the back end, so.
Yeah. I
And,
agree.
not screening everybody on a whole genome sequencing.
Right, right, which brings up a good point. We've thought about, should we, not report the conditions for which there's no treatment, but they cause, they may cause developmental disabilities, and wait until those kids have developed problems, if they do, and then, go back in and look at what the whole exome shows. So.
Thank you very much. And as a memento of your visit here, we also have another thank you plaque in purple, in a purple bag. Thank you, everyone.